Hello and welcome to MMI. In this series of tutorial videos, we'd like to show you the basics of the MMI CellCut and how to get good results for further research. The MMI CellCut is our laser microdissection system designed to cut tissues, large regions of interest or single cells, and even living cells for further processing. With its high-pulse UV laser for precise cutting and the patented CapLift technology for reliable tissue and cell isolation, it will help you with all your laser microdissection needs. In this video, we show you how to use fluorescence while working with the CellCut. You will also learn how to calibrate camera settings and add new fluorescence filters. If you want to work with fluorescence, don't forget to darken the room and turn on your fluorescence light source. CellTools comes with a number of predefined fluorescence filters which can be selected in the upper left corner of the software. Once selected, it's always good to check the camera settings. Notice that there's a special mode for fluorescence color imaging. You can also check the other camera settings to get a good picture. Once you're done, click on OK and the settings will be saved. If you want to implement new fluorescence filters, click on the pen symbol right to the drop-down where to select the filters. Click on the icon in the bottom left and the system will copy the current filter settings, creating a new entry that now can be edited. Type in the name of the new filter that you have installed, and then adjust the new filter number. That number is actually the position of your new fluorescence filter in the filter turret on your microscope. Unless you haven't installed any other, you can leave the number for condenser and camera as they are. Select a pseudo color for your filter if you want to make multicolor pictures. And again, if you haven't installed an additional camera, leave this setting as it is. Now you can draw in your shapes as usual. Select the Draw Shapes tool and just draw what you want to cut. Then just click on the Cut button and the system will start to dissect. In this case, the system cuts 2 times because we set the repeat value on 2. It is done and now we can collect the cutout. Some filters tend to decrease the laser efficiency. In this case, we recommend to do the drawing in the fluorescence mode and then switch to bright field for cutting. Draw your shapes as usual, then switch to bright field, and as you can see, your shape is still there. Then click on Cut and let the system do its job. We hope this video helped you with working with the MMI CellCut. Feel free to visit our other videos for more information about MMI instruments and handling.